coming to the objectives, at the end of this class, you should be able to describe the processes of oxygen transport in blood, draw a label diagram of oxygen hemoglobin dissociation curve, list and explain the factors that shift oxygen hemoglobin dissociation curve to the right and left, define oxygen saturation, oxygen content, and oxygen utilization. So there are only two main gases in the blood with respect to the respiratory physiology, which I'll be teaching you about, like how it's being transported by the blood. There is oxygen and carbon dioxide. Both of them are dissolved in small amount is dissolved in plasma, and the rest is carried in combination with in case of, for example, in oxygen, combination with hemoglobin, which is called as oxyhemoglobin. So hemoglobin is represented by Hb. So Hb plus O2 gives HbO2, which is the oxyhemoglobin. Now the portion which is transported dissolved in plasma is about 1.5%, which is a very negligible amount. So now this picture shows how it's being transported dissolved in plasma, which is about 0.3 ml of oxygen per 100 ml of blood. And in whole blood, it's transported in combination with hemoglobin, which is about 20.0 ml of oxygen per 100 ml of blood. So what's the role of hemoglobin in oxygen transport? Now each hemoglobin molecule, it has 4 parts. Okay, so like each part has a heme portion which has iron atom and a polypeptide chain attached with it. So now when each four parts, that is together, all the polypeptide chains of combined are known as globin, whereas the heme portion containing the part which is containing the iron atom is called as heme portion. Now each hemoglobin molecule can bind 4 molecules of oxygen, and this binding is reversible. So now when it's bound with oxygen, it's called oxyhemoglobin, and when it's detached from a hemoglobin, that is when it gives away the hemoglobin, it gives away the oxygen, it's called deoxyhemoglobin. Now if it is the ferrous ion of hemoglobin which binds oxygen, that is ferrous or Fe2 plus state is the only state in which the hemoglobin can bind with oxygen. Now if this ferrous ion it gets converted to ferric, that is Fe3 plus, it can no longer bind with oxygen, and when it's converted to Fe3 plus form, the hemoglobin is called as methemoglobin. So now in RBCs, there's a special enzyme called methemoglobin reductase, which reduces the methemoglobin back to hemoglobin, enabling the hemoglobin so that it can bind with oxygen. Now in certain cases, suppose this enzyme is absent, then the oxygen carrying capacity of the hemoglobin of the blood decreases, and it can lead to severe complications. So now 1 gram of hemoglobin can bind to 1.34 ml of oxygen when fully saturated. So the oxygen binding capacity of the blood is, assuming that the normal hemoglobin concentration is around 15 gram percentage, 15 into 1.34, which is 20.1 ml of oxygen per 100 ml of blood. Now arterial blood is normally 97% saturated with oxygen. Now coming to the oxygenation of hemoglobin. Now you have loading and unloading, 2 terms which I'll be introducing. Loading is the hemoglobin is loaded with what? With oxygen, and when it gives away oxygen, that is called as unloading. Now loading and unloading of oxygen is given by a simple reversible equation. That is, hemoglobin when it combines the oxygen, I told you there are 4 parts. So now when it combines the first part, Hb4O2 is formed. Now when it combines the second part, Hb4O4 is formed. And when it combines to the third part, Hb4O6 is formed. When it combines to the fourth part, Hb4O8 is formed. So now this oxygen binding shows cooperative binding. Now form the hemoglobin. I told you oxyhemoglobin and deoxyhemoglobin. Oxyhemoglobin, the normal heme contains iron in the reduced form, I told you, Fe2 plus. Now it is this Fe2 plus which has electrons and binds with oxygen. Now deoxyhemoglobin, when oxyhemoglobin dissociates to release oxygen, the heme ion is still in the reduced form. Now methemoglobin, I told you when Fe2 plus gets oxidized to Fe3 plus, it lacks electrons and cannot bind with oxygen. So the blood normally contains a very small amount of methemoglobin, and it is methemoglobin reductase, an enzyme present within the RBC, which reduces the methemoglobin back to hemoglobin, so that hemoglobin is now free to bind with oxygen. Now, what is carboxyhemoglobin? Carboxyhemoglobin is nothing but the reduced heme is combined with carbon monoxide. Now, carbon monoxide in a very excess amount, it leads to carbon monoxide poisoning. It is very common in people traveling, present in very crowded areas with lot of traffic, and in case of traffic inspectors, etc. So now the bond with carbon monoxide is 200 times stronger than the bond with oxygen. So what happens is carbon monoxide competes with oxygen to go bind with the respective sites in the hemoglobin molecule. So now when carbon monoxide goes and binds, oxygen can no longer bind with hemoglobin, and so automatically the oxygen saturation levels fall, and it leads to carbon monoxide poisoning. Carbon monoxide poisoning is a very dangerous condition because carbon monoxide gives a cherry red color to the blood, and so hence the paleness which is seen in case of anemic hypoxia or any other poisoning, or in case of hypoxia, there's the paleness of the skin. This is masked by the cherry red color. So the person usually diagnosing the condition is very difficult, and moreover carbon monoxide is an odorless, colorless gas, and so hence the symptoms which are present in the patient is also very is not very easily detectable. So now this table shows the amount of oxygen content and oxyhemoglobin, how much is present combined with hemoglobin, how much is present in dissolved form in the arterial venous blood. Now, oxygen physical solution, even though less, it's important because it's responsible for the tension of oxygen in the plasma. Now, it is this oxygen tension or the partial pressure which is denoted by PO2. The transfer of this gas, which enables the transfer of this gas from alveolar air to venous blood, that is from arterial blood to tissue fluid. Now, when hemoglobin saturation with the oxygen depends on the tension of oxygen in the medium. Now, when increased PO2, that is increased oxygen tension, more of oxyhemoglobin formation is enhanced. And when the PO2 decreases, that is a decreased oxygen tension, there is liberation of oxygen and reduced hemoglobin is formed. Now, coming to the saturation of hemoglobin with oxygen. Now, percentage saturation of hemoglobin with oxygen is given by oxygen content divided by oxygen capacity into 100. Now, the oxygen content is 19.3 ml per 100 ml of blood, and oxygen capacity is 20.1 ml per 100 ml of blood. So the percentage saturation will be 19.3 by 20.1 into 100, which is around 97%. So now coming to the oxygen hemoglobin dissociation curve. When the saturation of hemoglobin is plotted against PO2, we get the oxygen hemoglobin dissociation curve. Now PO2 is along the x-axis and saturation of hemoglobin with oxygen is along the y-axis. Now, hemoglobin saturation is the extent to which binding sites of hemoglobin are occupied by oxygen molecules. Now, the normal saturation of arterial blood hemoglobin is 97%, and saturation is determined by PO2. Now, oxygen hemoglobin dissociation curve is a sigmoid curve. It is not a linear relationship between PO2 and saturation of hemoglobin with oxygen. The curve is constructed by placing the blood in containers called as tonometers and filled with oxygen of known tension. After equilibration, the percentage saturation of hemoglobin with oxygen is determined, and the graph is plotted against different oxygen tensions or different PO2s. So now this is how it's plotted. You can see against this, as the PO2 keeps increasing, now we can obtain a sigmoid shaped curve. That is, as PO2 keeps increasing, the percentage oxygen saturation of hemoglobin also increases, but now only to a certain extent, after which a plateau is reached. The curve is sigmoid in shape. Now it has a steep face and it has a plateau face. Now, oxygen hemoglobin relationship is sigmoidal or S-shaped and not linear. The upper end of the curve is flatter than the lower end. So this is the sigmoid shaped curve. Now, it is seen that the plateau phase is very beneficial, and why the curve is sigmoid is that the plateau phase enables that, as if in case of conditions where the lung function is compromised or PO2 levels fall, the saturation amount remains the same. For example, around 60 millimeter Hg of PO2, the saturation is around 90%, and it remains so till 100 mm of Hg of PO2. Now, when in case of when the person ascends to high altitudes, or where in case pulmonary diseases, wherein the PO2 levels fall, it does not affect the saturation of hemoglobin, percentage of saturation of hemoglobin with oxygen. That is, it remains 90% for around the range between 60 millimeters of mercury of PO2 to 100 millimeters of mercury of PO2. So this is the basic advantage of the plateau phase. Now, hemoglobin oxidation dissociation curve, one important term you should know is what is the P50. Now, P50 is nothing but the PO2 levels at which hemoglobin is 50% saturated with oxygen. Now, increased P50 means there is decreased affinity of hemoglobin with oxygen and there will be more of unloading of oxygen. Whereas if the P50 is less, the affinity of the hemoglobin with oxygen is more, and so there will be more binding of oxygen with the hemoglobin. So now coming to the characteristics of the oxygen dissociation curve. It is sigmoidal in shape due to cooperative binding, that is increased affinity of hemoglobin for each successive oxygen molecule. The flat part is observed at higher PO2s in the lungs and the steep part is observed at lower PO2 in the tissues. A fall of PO2 from 100 to 60 millimeters of mercury does not cause significant reduction in percentage saturation of hemoglobin. At pressures less than 60 millimeters of mercury, the percentage saturation of hemoglobin falls steeply, causing greater release of oxygen to the tissues. Now, what is P50? I told you is the PO2 of oxygen at which 50% of the hemoglobin is saturated. At PO2s of 26 millimeters of mercury, hemoglobin is 50% saturated. At 40 millimeters of mercury, it is 75%, and at 60 millimeters of mercury, it is 90% saturated. So even if the PO2 of inspired air falls to 60 millimeters of mercury, hemoglobin saturation is not affected and the subject has no breathing problem. This explains why hypoxic ascent when hypoxia does not manifest even after ascending to 1,000 feet. Now, what are the factors affecting affinity of hemoglobin to oxygen? As cellular metabolism proceeds, more and more carbon dioxide, acids, and heat are all generated. So, as PCO2 increases, increase in H plus or decreased pH and increased temperature, the affinity of hemoglobin for oxygen decreases, and there's a rightward shift of the curve. That is, the oxygen hemoglobin dissociation curve shifts to the right. Now, this is the diagram which is showing you the normal arterial carbon dioxide levels and how the blue line is the leftward shift and the dark pink line is the rightward shift of the curve. Now, the presence of H plus ions and CO2 leading to unloading of oxygen by the hemoglobin is called the Bohr effect. Now, what is Bohr effect and its effect on P50 is the working range under resting conditions is 75% to 97% in the venous arterial blood saturation. Now, in exercise, this range extends from 35% to 97%. This shifts the ODC to the right, more oxygen is released to tissues, and so P50 increases. Increase in P50 indicates greater delivery of oxygen. Now, coming to another feature known as a 2,3-diphosphoglycerate or also called as 2,3-DPG. Now, this 2,3-DPG is an intermediate product of glycolysis occurring in RBCs, and it is formed as a result of anaerobic metabolism occurring in RBCs. As you all know, RBCs have no mitochondria, so aerobic metabolism, it does not depend on aerobic metabolism, and it instead depends on anaerobic glycolysis. And 2,3-DPG is an intermediary product of the embedded high Mayer-Hoff pathway, and where it's produced within the RBC. It's a byproduct of anaerobic glycolysis when glucose 6-phosphate is converted to pyruvate. It is present in especially high concentration in RBCs because of the content of 2,3-DPG mutase, the enzyme. Now, the affinity of hemoglobin for oxygen diminishes as the concentration of 2,3-DPG increase in the red blood cells. Now, increased 2,3-DPG, the curve shifts to the right. Decreased 2,3-DPG, the curve shifts to the left. So the curve shifts to the left, meaning more affinity of hemoglobin with oxygen, and curve shifts to the right in case of increased with DPG, that is decreased affinity of hemoglobin for oxygen, and there is unloading of oxygen by the hemoglobin. Now, people living at high altitudes show increased synthesis of 2,3-DPG. Now, at high altitude, there is decreased PO2. As an adaptation to hypoxia, there is increased synthesis of 2,3-DPG, which is due to the acclimatization effect. And 2,3-DPG binds to hemoglobin, preventing binding of oxygen to hemoglobin. Therefore, greater oxygen is released to tissues at lower PO2 levels. Now, factor shifting the curve to the left, that is decreased PCO2, decreased temperature, increased pH or decreased H plus ions, decreased 2,3-DPG, all shift the curve to the left. Now, there are two other factors which shift the curve to the left, that is the fetal hemoglobin and carbon monoxide. Carbon monoxide, I already told you, carbon monoxide poisoning, and fetal hemoglobin shifts the curve to the left. So that fetal hemoglobin has very high affinity for oxygen, and it can extract maximum amount of oxygen from the maternal blood and bring it to the placental blood and finally to the baby. Now, shift of curve to the left indicates less release of oxygen and greater saturation of hemoglobin with oxygen. Now, these are the two pictures which are showing the shift to the left and shift to the right. Shift to the right is increased PCO2, decreased pH, increased temperature, increased 2,3-DPG. Whereas in case of shift to the left, PCO2, decreased PCO2, increased pH, decreased temperature, and decreased 2,3-DPG. Now, this is what favors oxygen delivery from mother to the fetus. Fetal hemoglobin has alpha to gamma 2 chains. Now, poor binding of 2,3-DPG to gamma chain, as a result of which there is greater affinity of fetal hemoglobin for oxygen than adult hemoglobin, and the curve shifts to the left. This favors oxygen delivery from the mother to the fetus because fetal hemoglobin has very high affinity when compared to the normal adult hemoglobin to oxygen, and so a maximum amount of oxygen is removed from maternal blood and enters the placental blood and finally enters the fetus. Now, what is the role of myoglobin? Myoglobin is a hemoglobin pigment present in muscles. Now, in muscles, which is specialized for sustained contraction, example muscles of leg and heart muscles, the myoglobin is present in large quantities. Now, it contains only one heme group and one atom of iron per mole. Now, myoglobin takes up oxygen at a lower pressure much more readily than blood. Hence, dissociation curve is a rectangular hyperbola when compared to the sigmoid shape which occurs normally. Now, it does not show the Bohr effect, the myoglobin. It acts as a go-between in the transfer of oxygen from blood to the mitochondria within muscle cells. That is, the muscle cells, during intense isometric exercise, the muscles are contracting, the blood vessels are compressed as a result of the contraction, and the oxygen levels fall. Now, this myoglobin, when immediately it comes into action, it shifts the oxygen hemoglobin dissociation curve to the left. It has very high affinity for oxygen. It binds to the oxygen and immediately gives it to the very much needed muscle cells. Now, this picture shows the arterial end and the venous end. The arterial end, the PO2 pressure is 100 millimeters of mercury, whereas at the venous end, it's 40 millimeters of mercury. Then, as you all know, gas gets diffused from a higher partial pressure concentration to a lower partial pressure concentration level. So, oxygen is given out by the hemoglobin. And in case of carbon dioxide PCO2 levels are higher in the venous end when compared to the arterial end. So, hemoglobin gives out oxygen in the tissues and takes up carbon dioxide. So, this is arterial blood, and these are the percentage saturation and average oxygen content. Coming to the oxygen delivery, it occurs at rest, and oxygen delivery is the amount of oxygen delivered at the tissue level. Now, the oxygen delivery is determined by arterial oxygen content into cardiac output. The normal cardiac output is 5000 ml per minute or 5 liters, and the arterial oxygen content is 19.3 per 100 ml. So, when you calculate, you get 1000 ml or 1 liter per minute. Now, what is oxygen consumption? Our 5 ml of oxygen diffuses from the tissue capillary to the interstitial fluid. When 100 ml of blood flows, 5 ml of oxygen diffuses, and thus for total cardiac output of 5 liters or 5000 ml of blood, the amount of oxygen diffused will be how much? For 100 ml, for 100 it is 100 ml, it's 5 ml. So for 5000 ml or 5 liters of cardiac output, you get 250 ml per minute at rest. Now, what is the utilization coefficient? It is how much percentage of the oxygen which is delivered is utilized by the tissues. It is given by the formula oxygen consumption by oxygen delivered into 100, which is 250 by 1000 into 100, which is 25 percentage. Now, coming to the applied aspect, you have measurement of oxygen saturation of hemoglobin. Oxygen saturation is continuously measured in ICUs and emergency cases in the casualty, where you use a pulse oximeter which is attached to the fingertip or the ear lobule, and where pulsating vessels are accessible. Now, the red and infrared lights are transmitted through vascular bed from the pulse oximeter, and the pulsatile and non-pulsatile and total absorbances are calculated. Now, pulsatile components of the absorbance represents the arterial oxygenated blood. Now, you can also, I told you about the amount of oxygenation which is occurring in the human body and how oxygen is being transported. Now, oxygenation can be either in excess or it can be deficient. Now, when the oxygenation of the blood is deficient, it occurs in conditions which is called as hypoxia. Whereas in case of when there is excessive oxygenation, it occurs in cases of in cases when there is excess of metabolism and oxygen, when oxygen is reduced to water. Now, O2 is reduced to water. How it happens is it accepts electrons from the electron transport chain in the mitochondria. Now, oxygen accepts 4 electrons and it is reduced to water. Now, when it accepts less than 4 electrons due to some leak in the electron transport chain, superoxide, that is O2 minus, and hydroxyl radicals, that is OH minus, are formed. Now, these radicals are very dangerous, and when superoxide ion combines with nitrous oxide, you get peroxyl nitrate, which is also a radical, which is very dangerous and destroys the tissues. Now, how is this prevented? Is normally in the human body, your enzymes like superoxide dismutase, catalase, and reductases, which are antioxidants and destroy this oxidant radicals. So now, when these enzymes are deficient, the radicals are formed in excess, and the tissues get destroyed. And excessive oxygenation is actually bad for the body. Now, this is with respect to transport of oxygen. Now, coming to transport of carbon dioxide. Now, I told you about oxygen delivery, oxygen consumption, utilization coefficient, and the measurement of oxygen saturation of hemoglobin by a pulse oximeter. Now, coming to carbon dioxide transport. As you all know, oxygen is taken up by the blood as oxygen enters from the atmosphere into the lungs, and from the lungs it's transported by the arterial blood to the tissues. Tissues are the areas where the metabolism is occurring, and carbon dioxide is produced in large amounts. Now, this carbon dioxide has to be expelled out from the body. So, now how is this occurring? Now, how is carbon dioxide transported in the blood? Mainly in the form of bicarbonate, which is about 70%, dissolved in plasma, which is 10%, and in combination with hemoglobin, which is 20%. Now, H2O plus CO2, in the presence of carbonic anhydrase enzyme, carbonic acid is formed, which is H2CO3, and which is a very unstable compound and breaks to form H plus and HCO3 minus. So, carbon dioxide is transported in blood in physical solution and chemical combination. Now, the volume of carbon dioxide in the blood is arterial blood is 48 ml per deciliter, and mixed venous blood it is 52 ml per deciliter. Now, carbon dioxide is 20 times more soluble in blood than oxygen. 5 to 10% of CO2 is carried in physical solution, and most of the carbon dioxide is carried in blood in chemical combination. Now, chemical combination in the sense that is as carb amino compounds, either bound to plasma proteins or to hemoglobin, which is called carb amino hemoglobin. Now, as bicarbonate ions, also it is transported. Now, bicarbonate ions is the maximum amount in which carbon dioxide is transported in the by which the carbon dioxide is transported in blood. Now, coming to carb amino compounds, you have 10 to 20% of carbon dioxide carried as carb amino compounds in combination with proteins. Now, the RNH2 is nothing but amino group of the hemoglobin molecule. When combined with carbon dioxide, it forms RNHCOO minus. Then hydrogen ions are liberated. Now, hemoglobin is the most abundant protein and contains many NH2 groups. Now, when hemoglobin combined with oxygen, it's called carb amino hemoglobin as such because of the combination when it binds to the amino group of the hemoglobin. Now, reduced hemoglobin has a greater affinity for carbon dioxide. Now, coming to transport in the form of bicarbonate in plasma, 70 to 80% of carbon dioxide is carried as bicarbonate. Now, carbon dioxide plus water gives H2CO3, which liberates HCO3 minus plus H plus. Now, hydration of carbon dioxide in plasma is slow. Now, RBCs contain the enzyme carbonic anhydrase, which catalyzes the reaction of carbon dioxide with water to form carbonic acid or H2CO3. Now, carbonic anhydrase speeds up hydration of carbon dioxide by 13,000 times. Now, most of carbon dioxide released at the tissues enter RBCs to form H2CO3. Now, what is the chloride shifted systemic capillaries? At the tissues, carbon dioxide diffuses into the RBCs. H2O plus CO2 combines to form H2CO3, giving H plus HCO3 minus. Now, there is increased HCO3 produced in the RBC. Now, it diffuses out into the blood. RBC becomes more positive. Now, chloride is as such attracted into the RBC, and this occurs with the help through an exchanger, chloride bicarbonate exchanger or anion band exchange protein. The 3, which results in chloride coming inside the RBC and bicarbonate going into the blood. Now, this bicarbonate, once it enters the blood, it can be transported to the tissues. Now, this is called Hamburger shift, named after the scientist Hamburger who discovered this chloride shift. Now, this is chloride shift occurring at the level of tissues. Now, in lungs, just the reverse occurs, and that is called reversed chloride shift, wherein the bicarbonate enters into the RBC and chloride goes back into the plasma. And as soon as bicarbonate enters into the RBC, it combines with H plus to form H2CO3, and H2CO3 being an unstable compound breaks to release water and carbon dioxide, and it is this carbon dioxide which is expelled out from the lungs back into the atmospheric air. So this is the reversal chloride shift. Now, H plus release is buffered by combining with deoxyhemoglobin, and HbCO2 is formed. Now, this is the diagram which is showing the chloride shift. Now, as you can all see, CO2 plus H2O in the H2 forms H2CO3 in the RBC, and H2CO3 breaks to form H plus and HCO3 minus. HCO3 minus exits out of the RBC, and chloride enters into the RBC. Now, chloride shifted systemic capillaries, I told you the Hamburger shift. And now coming to the chloride shift, as a result of movement of chloride into RBCs, the total number of osmotically active particles increased, and as a result, osmotic pressure within the RBCs increased, and there is more absorption of fluid in RBCs. So, venous RBCs contain more fluid and they are bigger in volume, and hence PCV of venous blood is greater than arterial blood PCV, which is 3%, and venous blood RBCs are hence more fragile. Why? Because they have imbibed all the fluid and they increase in size. Now, coming to at pulmonary capillaries. At pulmonary capillaries, what has to happen? Now, this is the region where carbon dioxide has to enter the blood and has to be expelled from the lungs back into the atmospheric air. Now, this is occurring at the level of the alveoli. Oxygen enters the RBC. Deoxyhemoglobin gets converted to oxyhemoglobin. This has less affinity for carbon dioxide. Carbon dioxide is released and diffuses into the alveoli. Oxyhemoglobin in RBC is a strong acid H plus increases, HCO3 diffuses into the RBC, and it combines carbon dioxide to form H2CO3, and which splits to form H plus plus HCO3 minus, and H fluoride diffuses out into the plasma, and bicarbonate has in exchange for bicarbonate which is entered into the RBC. A reverse chloride shift in lungs is this picture is showing you how carbon dioxide is getting released back into the alveoli, and from alveoli during expiration it's going to be exiting out of the body. A carbon dioxide dissociation curve, like how I told you when PO2 is plotted against percentage saturation of hemoglobin with oxygen, you get a sigmoid shaped curve. Similarly, PCO2, and when plotted against carbon dioxide content, you get a linear relationship where the carbon dioxide content is along the y-axis and PCO2 is along the x-axis, and you get a nearly straight line with no steep or flat portions, and this curve is called the carbon dioxide dissociation curve, which is steeper than the ODC curve, and mixed venous blood carries more CO2 than arterial blood. Now, this is the carbon dioxide dissociation curve. Now, in case of at a given PCO2 level, the amount of the curve, I told you the curve at a given PCO2 level, when compared to the carbon dioxide content and the amount of oxygenation of the blood, so that is in case of deoxygenated blood, the curve will shift to the right. And in case of the same given PCO2 level, for example, 40 millimeters of mercury, at the same 40 millimeters of mercury, deoxygenated blood, the curve will shift to the downwards and to the and the curve will shift downwards and to the left. Whereas in case of at the same carbon dioxide level, in case of oxygenated blood, it will shift in the reverse direction. So now, this is with carbon dioxide. Curve, and this is an important effect which you should know is Haldane's effect, that which states that with increase in PO2 levels, it shifts the carbon dioxide dissociation curve to the right. So now this is with carbon dioxide and oxygen transport. Thank you very much.